Now we'll have Eleanor Dickinson talking about the role of deer and small mammals in the spatial distribution of tick vectors in a naturally fragmented landscape, implication for the transmission of Lyme disease.
Thank you. Hi, my name's Eleanor, and I'm presenting what I did for my master's project at the University of Glasgow with Roman Beacon and Caroline Millans, looking at how deer and small mammals affect the density of ticks and then how this might affect the transmission of Lyme disease. So there are many emerging infectious diseases which obviously cause a huge impact on human health. There's a lot that have emerged since 1940, about 300 or more, and they are, so vector-borne zoonoses, pathogens maintained by animal or wildlife hosts, and transmission to human hosts is what causes the zoonosis in humans, and this happens through infected arthropod vectors. And this makes it very hard to understand these infections and diseases because there's often multiple hosts that are involved in transmission and which have different contributions to transmission. This makes it hard to predict how infection risk changes across space and across different host communities, making it hard to research and understand where the risks come from. So host communities and disease risk play quite an important role together. There's a lot of work that's suggested that biodiversity can actually buffer the risk of infection to humans. So habitat change, which can reduce biodiversity and degrades habitats and wildlife populations, might actually lead to increased infection risk. So it's important to understand how these communities are working to try and work out how it's infecting humans. The theory is thoroughly debated, though, as the dynamics can be hard to test with different communities and diseases. So Lyme disease, it emerged in about the 1970s and has since been recognised as a very important infectious disease. It's present across the Northern Hemisphere, and there's a lot of disease in North America, but also it's found throughout Europe, including the UK. The infection is caused by the bacterial spirochete Borrelia burgdorferi sensu, which is a species complex of a lot of different genus species, 4 of which have been detected in the UK, and 3 of these can cause infection in humans. The disease is transmitted by ticks from the Exodus genus, which are 3 host life cycle ticks, so they need to bite a separate host to develop into each life stage, which they have 3, and then they must feed on a host to reproduce and lay eggs. The larvae do not gain infection from the adults through the eggs, so the larvae have to bite a host that's infected to become infected and then transmit in subsequent life stages to other hosts. This means that the nymphs are most important for transmission, so these are the ones that we've focused on during the study. And also the questing behaviour is where they're in the environment searching for a host, and this can be highly influenced by environmental factors. And so temperature and humidity and vegetation will influence how many ticks there are, and then also will have an impact on the risk of getting infected and being bitten by a tick. You also have multiple host species which are involved in the transmission of Lyme disease. They will have varying degrees of competency for transmission. Small mammals and birds are known to be the most important hosts for Lyme disease and for transmitting it. The most important ones in Europe are wood mice, voles, and shrews because they're very abundant and they've got a very wide distribution. So they are the main hosts for transmitting. And deer are also very important because despite being competent hosts, they feed a lot of ticks. So they will increase the tick population in the environment, which can then have implications for the transmission. So how does the different abundances and densities of different hosts affect the distribution of the pathogen and how it's transmitted? You have quite a complex cycle, which you start off, you tend to have a vector population in the environment which will have infected individuals. Even if one of these infected individuals were to bite a deer, they cannot transmit the infection through the deer, so you will not get any newly infected ticks. Although the deers can increase the tick population, which you then will have fewer infected ticks within the vector population, and then you get a dilution effect. However, if you then have competent hosts, and the more competent hosts you have, the more ticks they can feed, and they can also transmit the infection to new ticks, and so you'll have more infected ticks in the population, and therefore the pathogen will be amplified within the population, and you'll get more disease and higher infection risk. So our study, we were looking at the Loch Lomond area, which is the largest freshwater lake in the UK in Scotland, and we were using the island system as a way of looking at discrete habitat patches to understand what the different patches, kind of how they interact with disease transmission. And we also included 7 mainland sites for comparison. All of the sites tended to have woodland, so it's a good area for the host species and for ticks. And we also suspected that there were all the host species present at least some of the sites. So we expected to find mice and voles, although we didn't know which sites. They were definitely out on the islands. And there are definitely deer in the area, so roe deer and fallow deer are thought to be the most common in that area. So what we aimed to do was to quantify the cresting nymph density across the study space, and then to test whether this density varies as a function of deer density, the relative abundance of small mammals, and then also abiotic factors which affect cresting behaviour, so temperature, humidity, and then vegetation. And then we wanted to use this information to test how the communities can affect the prevalence of the pathogen and the distribution of it in the ticks in the study area. The methods we used a number of different methods to put it all together. Across the study site, we placed systematically placed transects, which were evenly spaced, and then at sampling points along these at each site, we used blanket dragging methods to collect the ticks. So dragged a blanket along 10 meters to work out how many ticks there are in that space, and would flip the blanket over and count the ticks and then collect them. For the deer density, we did 2 surveys, one in the spring and one in the winter, to walk along the line transects to count how much dung there was, and then also count the distance of the dung from the transect line using distance sampling methods. And we also had to mark the dung to calculate the decay rates, so we could estimate how many deer there were producing how much dung we counted, so we could estimate density using distance software. And then looked at the small mammals. We used a hair tube sampling strategy at the same places that we conducted the blanket dragging, and just put sticky hair tubes out, which were optimized size-wise for the different species of small mammal, and then baited them, so we had the small mammals going in and leaving hairs, so we could find which small mammals there were, and then identify the hairs, which is currently being done. So what we found was that we had questing ticks everywhere. The density ranged from less than 0.05 per metre square to over 2.5, which was quite a big difference. We didn't see any significant difference between the mainland and island, although the islands did show a higher degree of variation between the sites compared to the mainland, where there wasn't as much difference between the different sites. And then the deer density, we found that there was also no significant difference between the mainland and island, but again, there was more variation on the islands. However, during the different times that we did the surveys, there were a higher number of deer on average during the winter than in the spring, and this was also more extreme on the mainland sites. So we think maybe the deer were moving further away to be able to eat more food during the winter, during the spring, sorry. And also the small mammals, we weren't sure exactly how many we'd find on the islands, but we found small mammals at every site, which was something we weren't expecting. And the relative abundances that we found were more extreme again. So we went from 20% to 83% of the number of tubes that were occupied, and it was much more extreme on the islands, although there was no significant difference between the mainland and island. So how does this affect the questing nymph density? We found that deer density did significantly influence the density of questing nymphs, with a significant increase with deer density. And the relative abundance of small mammals didn't affect the questing nymph density at all. The other significant variables of questing nymph density was the temperature and humidity, which had a significant interaction. So the more humid it was, the more ticks there were, and the cooler it was, the more ticks there were. So that obviously massively changes how many ticks you're going to find in the environment. And then also the vegetation height and density affected it. So you can see on the graph of the deer density that the lines show that at lower densities you're more likely to find more ticks, and at the higher densities you'll find less ticks. So what implications might this have for the transmission of the pathogen? We know that there are potential hosts for the Borrelia at each site, so all the small mammals, and from observations we can see there's lots of birds, which are also important hosts, but we couldn't quantify them at the time. So we know that the transmission of the pathogen is not going to be limited by those factors. However, we hypothesized that the deer density might be a significant predictor of prevalence in the ticks. So we think that at the lowest densities of the incompetent host, that you'll have a lack of host effect, you'll have less ticks, and then less chance of having high prevalence of infection. But at higher deer densities, you're likely to get the amplification effect that I mentioned earlier, where you have more ticks in the environment and you have the hosts that can transmit the infection, and so the pathogen will be amplified. But as you get to the more extreme, highest levels of deer density, you will then get the dilution effect because you have the larger tick population, but not enough hosts to maintain the high levels of transmission. So next, what we need to do is, we know that there is the pathogen present in the study area. The map shows a few locations where ticks that were collected in 2015 showed had infection. So we need to test the ones 2016 so we can compare the different sites with the different estimates of deer density and small mammal abundance. And then we can hopefully understand whether the deer or small mammals actually do influence transmission of the pathogen, or whether it's driven more strongly by other factors associated with the fragmentation of the habitat or spatial isolation of the smaller island sites. Thank you.
Thank you. Thank you very much. We have time for some questions. Yes.
I just wonder how the Boulinier gets onto the islands. Are the livestock moving back and forth between the mainland and the islands?
There's not livestock, but there's definitely birds that travel between the islands, and also the deer can as well, so they maybe move the ticks so the ticks can stay on them when they swim between the islands. Yeah, so it does get on somehow. We're not exactly sure the way, but it's mostly birds and deer, I think. Yeah.
Any other question? Maybe I have one just to follow up on this. How do you think the local populations are isolated, are closed, or do you think in terms of numbers, like bird could bring infected ticks, but could they bring like a female that will lay eggs locally, and could this influence the dynamic strongly, or is it more independent populations between the islands, for instance?
Yeah, I don't think that could happen much with birds bringing adults, because you're more likely to get larvae feeding on the smaller hosts. But the deer definitely could bring adults, which would reproduce. And yeah, but then like small mammals, we think that it's very isolated populations and they don't transport much. So there is some degree of movement between the islands, but not a huge amount, and we don't actually know how much. Yeah. But.
Any other question?
Oh, sorry.
Yeah, they're coming. Yeah.
Ooh.
Andy?
So you lumped all the small mammals together. I just wonder if you've got a feel for whether one species is more abundant or contributes more to.
So we have an honours student at the moment who's looking at the hair tubes to try and work out which species there were. And it's a fairly hard thing to do, but we can definitely distinguish whether it's shrews or mice or voles. So we're going to get a little bit more information on that. Yeah.
Yep.
I just wondered, when you're using the blanket, so you're saying in high density growth you didn't get as many ticks.
Yeah, and the
Is that
density of
definitely because
vegetation.
of high density growth, or would it be like a sampling error if your sampling technique isn't as effective?
Yeah, it could be that. There's definitely, I think there's quite a lot of work that's been looked into how accurate actually blanket dragging is, because obviously it depends so much on local, like how many like different variables locally, and obviously the temperature and stuff, it depends how many ticks there are, but there's going to be more actually in the vegetation. And yeah, if it's really dense vegetation, it might be harder to pick as many up, but it's quite hard to account for that. You just have to kind of keep it in mind.
Thank you. I think we can have a round of applause again. Thanks. Can you put it full screen?
Full screen is
Okay, now we'll we're going to have Hashas von Hardenberg, who will talk about higher risk of gastrointestinal parasite infection at lower altitude, suggest possible constraints on the distributional niche of alpine marmots.
Yeah, thank you. I have to make a little disclaimer. I have a bit of an imposter syndrome. I'm actually not a parasitologist. I'm an ecologist. But the other people, Stefania Zanetti in particular, who is the first author, she is a real parasitologist from the University of Torino, with whom we collaborated with the University of Chester, Gran Paradiso National Park, where this study was done, and University of Torino, who did this actually. Why alpine marmots? Alpine marmots, well, everyone knows them. They're really interesting and funny species, actually, very social species. They live together in groups, in family groups. And they live in high altitude. So the distribution is bounded. The altitudinal niche is bounded about between 1,700 and 2,800 meters above sea levels in the Alps. That's where we find marmots. They're highly adapted to this kind of environment. They go in hibernation in winter, and this, of course, causes a lot of costs. They have a cost for hibernation. They need, during summer, to accumulate fat. That's the main occupation during summer, to be able to survive this very stressful hibernation process. If you didn't notice, there is a marmot, actually, even if you couldn't see it, it's over there. So gastrointestinal parasites are possibly a high cost for alpine marmots as they deplete resource which otherwise would be available for fat accumulation, so which could be used for hibernation. So this brought to us to our main hypothesis, where we wanted to find out if at higher altitude, there is a lower risk actually to be infected by parasites than by gastrointestinal parasites than at lower elevations. And if this is true, of course, well, this is a hypothesis which are testing here and I present here, but this brings forward some interesting ideas, like it may well mean that parasites could contribute in actually contributing as one of the factors which restrict the distributional niche of alpine marmots. Where did we do this? We did this in the Gran Paradiso National Park, which is in the northwest Italian Alps. It's the oldest national park in Italy. It was founded in 1922. And well, just to tell a bit of history, our study area where we are studying this, what did I do? Okay. Is Orvieres, this area here. This is in beginning spring, this is in fall, in September, at the end of the season, in which the marmots were out. And this hut, actually, the hut of Orvieres, was a royal hunting hut, which a king of Italy used in the 1800s, when the Gran Paradiso was still a royal hunting reserve. He used it to hunt ibex, actually, in the Gran Paradiso National Park. So what did we do? Well, we decided to choose, this is a long-term study we're following in the Gran Paradiso, so we know very well the marmot families in the Hula area. Many of them are actually tagged, individually tagged, so we have a good idea of the families in this group. And we choose 22 families along an altitudinal gradient. So we went from basically pretty much covering the whole altitudinal range of marmots, going from 1,600 meters to over 2,800 meters. And we choose 22 latrines inside the burrow systems. They have usually a latrine which can be quite easily identified, each family, where they deposit feces. So we could repeatedly during the whole season collect feces from these latrines along this altitudinal gradient, where we had, as I said, 22 different families which were following. So we had these 22 latrines following up there, and we collected a total of 587 fecal samples from the beginning of season when the marmots come out from hibernation to September, and collecting between 2 and 46 fecal samples per site. 2 is just one case where we had just 2. Most groups we had a median of 27.5 fecal samples collected over summer for each latrine. What did we do with all these fecal samples which we collected? well, I come to that in a moment. We also, for these sites, we have also information, besides the elevation, we have information about, about solar radiance, because solar radiance can affect the survival of parasite eggs in the field when they are released in the field. And to make sure that we checked this, and actually there's no correlation between elevation and solar radiance. So we have two quite independent variables which we could include in our models and see the effect of both of them without any problem of collinearity. My parasitological friends had the honor to analyze all these species, and we looked at Ctenotenia marmota, which is, and Ascaris levis, the two typical parasites in alpine marmots, and they counted eggs and oncospheres and oocysts actually in the fecal samples. So this is also nice, and if you look at the prevalence of different of these indicators of these parasites in the fecal samples, we see that there is a lot of cases in which actually we don't, there's a lot of cases in which not always, we have a lot of absences basically in our fecal samples. No? So, and it's well known that in fecal samples we have a high rate of false negatives. So we have definitely a problem of what me as an ecologist would call imperfect detection. This is actually very similar. So how do we solve it? So we cannot just take this data because we have this problem. How do we account for this imperfect detection? This is actually very similar to the situation which ecologists usually have when we try to analyze occupancy of animals in the field, and we want, and of course, sometimes we go in a site and we don't see the animals. So, so what we did, we used actually what I called site occupancy models, which are usually generally used in ecology, and I'm not aware of any application of this actually for specifically for gastrointestinal parasites from feces. They have been used in parasitology, but not in this specific field. Site occupancy models are very good to disentangle occupancy, so it's a probability that actually the parasite is present in a specific site from the detection probability. So we considered the feces collected in the same sites as repeated observations. And for example, we could have 4 feces in one site in which we have, in one we find it present, in 2 not, and 1 not. So in 2 it's present, and in this sample occasion HI. And we can basically calculate the probability that this situation happens. And this is another case in which actually we don't find any. So in in this specific site we don't find any eggs. But this may mean 2 things actually. It may mean that the parasites are present, but the eggs are not found simply, so we have a problem of detection, or really that the parasites are absent. And we can still model this because we can add the this is the probability that the species is actually absent. So we can add this, and we can, I'm not going too much in the mathematics here, but basically we can, out of this we can get a joint likelihood by which, with maximum likelihood, we can get, actually we can extract, combining the product of all our sites, of all our probability for all our sites, we can get the likelihood, and we can get the probability of the occupancy and the detection probability. So basically we can split, we can disentangle the two, which is really good. We did this using a commonly used package, unmarked in R. I'm almost there. And the literature, the gurus of occupancy models tell us that actually when heterogeneity in detection probability is a result of variance in abundance, we can actually use detection probability as a proxy of abundance. And this is, seems to me, actually the case of parasites in feces. So this is what we got. Sorry, this is not so clear to see. Oh, sorry. So basically, the first up in the right corner there is actually the elevation, is the detection probability against elevation. So indeed, we find a decline in detection probability, so as a proxy of abundance, with increasing with increasing elevation. And this is a radiance. This is for Ctenotenia marmotae. As well, with increasing solar radiance, we have a decrease in the probability of detection. And this is a Julian date. We have a similar for Ascaris levis. In this case, in the model, the solar radiance didn't enter the model, was not important, but again, here we find a strong effect of altitude on the detection probability. And this is our model selection process. So the two models, one for Ctenotenia marmotae, the best models always have basically a constant occupancy. And that's a point, little point you find in the parentheses. And while we have a probability, a detection probability, the best model for Ctenotenia marmotae is where we have altitude plus date plus solar radiance, and for Alevis we have just altitude and date. So conclusions, 1 minute and perfectly on time, incredibly for me. And so we detection probability as a proxy of abundance declines indeed with elevation. And we can talk about what this means and what implications this may have for marmot biology. And I think it shows relatively nice that site occupancy models can be a useful tool to model abundance of gastrointestinal parasites when we're using non-invasive coprological analysis of fecal egg counts. Thank you very much.
Thank you. Any question? Yeah, Romain.
I'm just curious. I wonder how the composition of the groups change with altitude, and how do you have differences in the size of the groups between lower and higher altitudes? And does the composition change during the season?
No, during the season, no, because these are stable groups. These are stable family groups in marmots. So usually there's a couple of dominant marmots, male and a female, and the yearlings and the pups, basically of the marmots, they stay in the family group until 3-4 years of age before dispersing, and they basically stay in the family group as helpers with the pups of the year. So you can have quite big family groups. In our situation, we didn't notice, I should look over time, we have 10 years of data now, if there is a difference in the composition. I expect actually that survival is actually lower at low altitude, but this is data I haven't analyzed yet. So yeah.
Thank you. Okay, yeah, Heike.
What are the intermediate hosts for these parasites? Are they,
Yes.
are they soil
The Alevis is a soil parasite, while the Ctenotenia marmotae has orbital mites as a intermediate parasite, intermediate host.
So the abundance of mites declines upwards?
Sorry?
The abundance of soil mites declines upwards?
I don't have this information, but yeah, sure. Yeah, sure, it's related to that. Yeah. But that's why we looked at two different kinds of parasites with two different life histories to see to avoid that this could be true. And we have consistent results for a parasite where actually without an intermediate host and one with mites as an intermediate host, and the results are very similar between the two.
Thank you. Maybe we'll stop there. I had a question,
Yep.
but I'll keep it for you after.
Yeah.
Yeah, we don't have much time.
Okay.
Well, just it's a nice application of the method. I'm just wondering whether, in terms of number of replicates by date and so on, did you feel any constraint, or do you have any advice?
The more the better, but with 3-4 usually should be enough, exactly like for any other occupancy model, actually. But of course, the more you have here, we had quite a bit actually, but besides some sites which actually we had to remove because we had too few replicas. We had one site where we had just basically 2 data points. Yeah.
Okay, thank you. We'll upload again. And now we'll have a talk by Joan Lillo, backseat drivers, microparasite alter ontavirus dynamics by changing host demography.
Thanks. So I'm going to talk to you briefly today about a part of a Marie Curie fellowship that I undertook in Italy just recently, in Trentino, in the Fondazione Edmund Mack, having a leave of absence from my day job in Cardiff University. This works on a data set that was collected by Adam Konechny, a previous Marie Curie fellow, and it's in association with Heidi Hauff, who was my supervisor, and Roberto Rosso is a mathematician over there, and Giovanni Marini. The project was called Paracourt, and it stands for the role of macroparasite co-infection in rodent-borne microparasite transmission. Wild rodents, as we've heard already in numerous talks, are great hosts for lots of different parasites. And they carry a lot of human transmissible diseases, including hantavirus, LCMV, tick-borne encephalitis, and the list goes on. I'm going to speak to you mostly about hantavirus and LCMV today. But they also are hosts who a lot of endemic macroparasites, as are most natural species, including ectoparasites like ticks, fleas, mites, and endoparasites, various helminth species in particular. And this is interesting from the perspective of co-infection because we know that, broadly speaking, micro and macroparasites drive different arms of the host immune response. So very broadly speaking, macroparasites are known to push a more Th2, T helper cell 2 response, and microparasites more of a T helper cell 1 response. And that provides a mechanism of potential interaction between these species because we also know that there is a rough seesaw between these two responses. So you can't have a very high Th2 response and a very high Th1 response. They tend to push, they have actually a co-down regulatory effect upon one another. And that is the majority of what my previous work has looked at as in-host interactions, either through these immunological mechanisms or through, or through resource competition between parasites. But what I'm going to focus on more today is a less well-documented method of interaction, which is very indirect and is through the host demographics. To my knowledge, there's only one paper by Andy Fenton that has looked at this theoretically, combining the classic models of Anderson and May for the SIR and the macroparasite model, and shown that through demographic changes in the host population, you can have interactions between your parasites. And just to give you a very, very basic example of what I'm talking about here, if we imagine these blue dots are a host population, and our red dot is an infected host, and this infection passes through the population over a period of time. Now, if you imagine we start that population again with a second parasite species in here, and I'm going to show you an extreme example, but this second parasite, the yellow parasite, kills off some of those hosts. So over the same period of time, the red parasite cannot spread through as many hosts simply because it has not got as many hosts to spread through. And so we see different dynamics of the macro and of the parasite, in singly infected and co-infected host populations. Now, that was an extreme example, but of course, that principle can apply with just any changes in the demographics of the host population. The data that I used was by, as I said, for Adam Konechny, a prior Marie Curie fellow who worked on this data set called Rocco Alps. I won't go into what Rocco Alps was about, but his data was 2 years of data from 2011 and 2012 between April and September. It was taken from 2 different altitudes, high and low altitude, so cooler and warmer, at 8 different trapping sites within those different altitudes. It's marker capture data on yellow-necked mouse and voles in the main, in bank voles. There were other species in there at much lower numbers, and we collectively call them other species, of small rodent, obviously. We collected data on the species, the mass, the sex and sexual status, and from that we can calculate the proportion of females to young, etc., the ratio of different species. We also have fecal egg count data for Helimusmoides species, different species in the the voles and the mice, and Rodentolepsis, which is a tapeworm, and also for coccidia. And then we have counts also of ticks, and then presence-absence data, because there's so many of them, for fleas and mites. And then similarly, we also have antibody presence-absence data for hantavirus and LCMV. Now, both of these viral infections are known to be chronic infections. So once we saw an animal had been infected, there's a detection error issue there, of course, but across the board, but once it's detected, we assume that animal is then infected for its lifespan. And we asked a number of questions. Firstly, what controls the host demographics in this population? Then, turning that on its head, what effects do the host demographics have on the parasites in the population? We looked at that internal interrelationship between the parasites. What do the parasites interact with one another within the host? And bringing that all together, the main aim of the project is how can we best control both parasites and host dynamics at the same time. The hosts are doing damage to agriculture, etc., carry human transmissible diseases, so people want to control them, but we don't actually know what happens to the parasites in this circumstance. I'm not going to talk to you today about those latter two, that's just to say what the overall project was about, but I'm going to concentrate on this control of host demographics versus the host, what the host demographics does to the parasites. And we did this in a series of mixed models using the package AS-REML in R, controlling repeated measures for animals, the grid they were captured in, and their year of capture as random effects. The demographic parameters that we used in our models, we calculated using a package called RCapture in R, and we separated them out by grid, by year, and by session, and session is basically month, with a tiny overlap on a couple of months. We had also mean parasite prevalences, as I showed you before. These were all highly correlated, all of the parasite counts, some negatively, some positively. So rather than trying to put them all in a model, which would have been non-legitimate, or trying to deal with them individually, I did a principal component analysis on these. And what we see here, I hope, is that we get a fair explanation of the parasite load with just the PC1 axis. 45% of the variation is explained by PC1, and you can see, I hope, in particular, that the hookworm and the ticks and mites separate out on the PC axis there. So one hookworm loads negatively on PC1, and ticks load positively. We then did a series of GLMs producing from the demographic data, using these three parameters as demographic dependent variables. The host survival, calculated from R capture, and abundance, also calculated from R capture, obviously quite interrelated factors. And then we didn't have a good measure of fecundity, so we used subadult, proportion of subadults in the population as a proxy for fecundity in the population. Our independent variables were session, which is that sort of month variable, altitude, high or low, and then the proportion of other hosts that were there. So we have the proportion of voles and other small mammal species. Principal component 1 was then included as our measure of parasitism. And grid and year were included as random effects, as I mentioned. The first thing that comes out, I'm going to only show you the data for the mice because we had more data for mice, and so the statistics are robust. What I'll say about the voles is we see basically the same pattern in the voles if we do the analysis on the vole population, but it's not as strong a statistical evidence. So we find, firstly, that the proportion of voles in the population is strongly negatively correlated to the survival of mice, and we see the same sort of thing if we reverse that for the voles, but this on abundance. And we see this, although obviously we can't do it so strongly within individual grids, but we can see the same trend within individual grids, so it doesn't seem to be just a case of some grids are better than others for these species. But more importantly, more interestingly for us is the parasite information. This is a bit of a complicated graph, so I'm going to spend a moment on it. What you see on the y-axis is mouse survival. The two lines, the colours, indicate high and low altitude. So if you think blue is high, cold, and red is low. And at first glance, it might appear to say that they have different effects of parasites at different altitudes, but I don't believe that's what's going on here. And if I can remind you that if you are on the left-hand side of the PC1, on the negative side, you have high numbers of Helicomosmoides. If you're on the far right, you have high numbers of ticks. So what I believe is going on here is the same thing driven by different parasites at different altitudes. So macroparasites are driving a reduction in survival. So if we look at the blue line, as we get up at higher altitudes, as we go higher numbers of worms, we get lower survival. At the low altitudes, as we get higher numbers of ticks, we get lower survival. And in fact, if we just look at the sort of mean numbers of those parasites at the two different altitudes, you see that you have far more worms in the higher altitudes than you do in the low, and far more ticks in the low. So to summarize, what I'm saying is that the macroparasites are driving down survival in the mice. Whether that be at different altitudes, one of the parasites is more important than the other. We then turned that question on its head and said, what do the effects of the host demography have on the parasites? And I think I'm running out of time, so to cut a long story short, what we find is that there's no effect on the macroparasites of the mouse abundance, but the abundance is strongly and significantly related to the prevalence of both LCMV and hantavirus. And what we see is a strong dilution effect, such that as mouse abundance increases, hantavirus goes down. Okay, quite substantially down. And we can actually see this very clearly in the raw data. In the first year, in 2011, there are very few mice, and the prevalence is, is considerably higher, is tenfold higher than in the second year when we have far more mice. So overall, what this is drawing that together, what we're saying is that we have competition, it seems, between these two species, and that drives mortality. We have parasites, which are also driving mortality, and so we are reducing the abundance of the population. The viruses, the two viruses, actually increase when the population goes down. So we actually have this indirect effect where the macroparasites can be reducing host population and driving up the prevalence of hantavirus. At the moment, we are now completing this work. We've created a model of this system, and we're currently fitting it to the data. So we're at the last stages of fitting it to the data, basically, with the plan to be able to manipulate the system and say, what if we change the number of parasites? What would we expect to see in the prevalence of the prevalence of the LCMV and hantavirus? I think I'll leave it there. Thank you very much.
Thank you. Any question?
In the plot where you had the high and lows, you had the macroparasites, it's the worms and the ticks. So here you've got lots of worms and here you've got lots of ticks. In the middle of those animals actually got a smaller number of each, but a lot together. So what I'm asking is, what's the total number of macroparasites, no matter what their type is?
It's not quite as simple as that. If I actually do this analysis using those two parasites, what we see is just individually in the model. What you do see is that the significance is not as high because there's a joint effect of the two parasites clearly. But what we do see is that over the whole range of altitudes, hookworm is important. Helicomosmoides is important, whereas ticks are only important at the lower altitude. So as you go up that line, you basically lose ticks quite quickly.
Any other question? Paul?
I was also wondering, is there any kind of expanding or collapsing populations over the years, so that if it expands, so it leaves behind the parasite, so you get low incidence, for example?
Sorry,
Are these populations stable, or are they expanding, for example, that they leave behind the parasite?
I don't know whether they're expanding spatially, but in terms of year to year, there's massive differences in the dynamics year to year. So in 2011, we actually had very few mice all-voles. And in fact, if you looked at the number of juveniles, subadults per female, it was about half a juvenile per female, where it was 20 juveniles per female in the following year, and a tenfold increase in the population. So there are definitely massive dynamics. We don't see the data set's too short to see any die-outs, but from of the parasites we looked at, they're not dying out completely, but there are these severe fluctuations depending on what. Not to go into too much more detail, but there is some evidence from what we've looked at now that the subadults are what's driving that apparent dilution effect in the hantavirus, because the subadults in the first year, the prevalence of hanta is the same in both the subadults and the adults. In the second year, it's much lower in the subadults when there's this influx. And we think that's just breaking up the social networks, basically.
Thank you. Okay, we'll go to the next one. Felicity Wine will talk about detection of Rana virus and Batrachochitrium
Chytrid, it's fine.
BD in northwest Costa Rica.
Yeah, so thank you. This is part of my PhD project at Plymouth University, done with my supervisors Rob Puschendorf, Steve Price, and Mary Knight. So global enigmatic amphibian declines were first officially recognised in the late 1980s, and since then amphibians haven't been doing much better. 32% of amphibian species are thought to be threatened with extinction around the world. And in Costa Rica specifically, we know that 23 species suffered serious declines or went extinct entirely. And that occurred in and after 1987. And these events were linked to both disease and also climatic events. And those species that were stream associated or occurred at altitudes above 500 meters or of certain taxonomic family were more likely to be affected. But recently there's been a bit more of a glimmer of hope. Some of these previously presumed extinct species have been rediscovered in new populations. So the amphibian chytrid fungus, Batrachochytrium dendrobatidis, was first discovered in the late 1990s, and it's a skin fungus that causes an electrolytic imbalance resulting in cardiac arrest and death in amphibians. And this has been linked to the extinction and steep population declines of over 200 species worldwide, including those in Costa Rica. In addition to chytrid, there's also another pathogen, Rana Virus, that's been linked to amphibian population declines. And here in the UK, Rana Virus has been associated with declines of up to 81% in UK common frog populations. And in Spain, more recently, it's been found that this virus has been associated with the decline of multiple species. It's also associated with reptile and fish die-offs. And amongst many causes, it can cause death by organ necrosis. And it's been very recently detected in Costa Rica from samples that were taken in 2007. And as a result, we don't really know anything about the virus here. So my initial aims were firstly to try and do a population health check for both the amphibians and reptiles, just to see what was where, and also see if we could detect rhinovirus in my field site. And if so, what the distributions, both host and spatially, for this and chytrid were, and whether there were any environmental factors that were associated with these distributions. So my field site is in the Area de Conservation Guanacaste, or the ACG, and we know that at least 8 species of amphibians declined or disappeared here, which is about 15% of the total species found there. And for Costa Rica, there's been one distribution model that was estimated for chytrid, which is on the slide here. And the circled area is where my field site is. And this distribution model predicted that the areas in blue are very low prevalence. These are the sort of dry forest areas and very low elevation areas. And the areas more likely to contain chytrid were those higher altitude areas in red and yellow here. So this led to the hypothesis that these dry forest areas provided refuge for amphibians from chytrid. But for the ACG, we have almost no data for this. There's been one survey for chytrid here, and this was only in the dry forest. And we also have absolutely no data for rhinovirus here either. So to collect that data, I sampled live amphibians by swabbing their skin to try and detect chytrid, and toe clipping them to try and detect rhinovirus. And I also used samples from road-killed amphibians and reptiles just to try and detect rhinovirus, and these were usually liver or other internal organs. And all my samples were collected from dry forest or wet forest sites, but for my roadkill samples, they were mostly taken from the dry forests, as this is where the roads were. And in total, I had 6 sites, 3 of which were wet forest and 3 of which were dry forest. In addition to collecting samples from the present day, I also tested 13 samples of 5 species for rhinovirus. And these samples were collected in 1987, so just about when amphibians were starting to decline. And they were collected from within my field site in the ACG. And what's interesting about the species here is that every single one that we tested either disappeared completely from the ACG, every single one disappeared from the site they were collected at, and every single one experienced some form of wider decline within Costa Rica. So to try and detect chytrid from my swab samples, I used standard protocol qPCR, and for Rana virus, I used a nested PCR and gel electrophoresis. And I also wanted to try and determine whether there were differences in prevalence between our wet forest sites and dry forest sites, so I used chi-square to do that, and wanted to see whether there were any associations with infection likelihood, environmental associations. And so I used a logistic regression, and the factors I will focus on here are temperature and taxonomic family. So I sampled, most of my samples came from last year, and we had a horrendous El Nino. This resulted in about 95% less rain in my field sites, in my dry forest sites. Really not great for frogs. However, it means we have quite a unique baseline data set from an El Nino. And from this, we found that 19.4% of the amphibians we sampled in the wet forests were infected with chytrid, and that was it was detected in 11 species. And we actually only found chytrid in the wet forests. We also found that certain families were more likely to be infected, and these were the glass frogs, the tree frogs, and the true frogs, so Centralenidae, Hylidae, and Ranidae. And this pathogen's occurrence was also associated with temperature, whereby our hotter sites were less likely to contain chytrid than our cooler sites. For Rana virus, we found an overall prevalence of 14.5% amongst all sites, and that was from 17 amphibian species and 4 reptile species. And 12 of these amphibians and all 4 reptile species, which included pond turtles, snakes, and lizards, were previously unknown to be hosts for Rana virus. Unlike chytrid, we found Rana virus at every single site we surveyed at. But again, more like chytrid, certain families were more likely to be affected. Here, the glass frogs and the true frogs, as with chytrid, and also the microhylids. We also found an association with temperature, whereby the hotter sites this time were more likely to contain Rana virus, which is the complete opposite to our chytrid data. And we also detected Rana virus from our historical samples. So 5 of those 13 samples were positive for this pathogen. And this indicates that Rana virus was present in Costa Rica right before amphibian declines. So overall, we detected widespread infection of Rana virus from all of the sites we sampled, wet and dry forest. And for chytrid, we only detected it in these wet forest sites. We failed to detect it in the dry forests. And the previous survey I mentioned about, the only one that had been undertaken within the ACG, actually did detect chytrid in the dry forests. And we sampled in the exact same sites and had about the same sample sizes, but still failed to detect the pathogen here. So that's led us to kind of think that maybe this El Nino that's provided very unsuitable conditions for chytrid has led to the reduction in the prevalence of this pathogen here. And this also supports the idea that these dry forest sites don't contain much chytrid and could be acting as refuges for amphibians against this pathogen. However, we found the exact opposite for Rana virus, whereby our dry forest sites, these refuge sites, had exceedingly high levels of Rana virus in comparison with our wet forest sites. And that's of concern not only because these populations in general have suffered declines, but also these refuge sites contain species that occur nowhere else in Costa Rica, and they used to be widespread. And we've also detected Rana virus in those species. So we don't actually know what impact that could be having on them. It's slightly worrying that this opposite trend for chytrid and Rana virus, this temperature trend is in the opposite direction. And this means that, at least from our sites, there is no refuge site for both pathogens, both of which obviously have been linked to population declines elsewhere. It's also slightly worrying that Rana virus seems to be increasing in warmer conditions. So elsewhere in the world, we know that the prevalence for Rana virus is higher in summer months, in the warmer months. The impact that processes such as climate change might have on these sites that contain threatened species is of concern. However, we found loads of infection for Rana virus. We didn't find much disease. So this guy was the only animal we found that had any visible signs of Rana virus disease. So I don't know if you can see, but on his leg there, he has some red areas of hemorrhaging. He also had loads of chytrid, so we don't know what killed this animal. We know that Rana virus has been present in Costa Rica for 30 years now, so it could be that these populations have already adapted. I mean, we don't know, we know it's been in Costa Rica for 30 years, it could have been there for longer. So we really have no idea if this is any sort of conservation threat yet, but we do need to know. So I also said about there not being many surveys undertaken in my field site. So what I'm trying to do at the moment is gather more information on what actually is normal for these sites, whether this high Rana virus prevalence is normal or not. So that's what I'm working on at the moment. I have some data from this year, and obviously I'll need subsequent data to establish that eventually. And I'd like to end just by summarizing. So we know that chytrid and Rana virus have the capacity to cause amphibian population declines. We knew that chytrid was present in Costa Rica and very strongly associated with the amphibian declines there. We now know that Rana virus was there at the same time. And both pathogens seem to be correlated with temperature in the opposite direction. And this means that amphibian populations may struggle to avoid infection by either or both pathogens. So thank you very much for listening, and I'd like to thank everybody who helped me with this part of my research. So thank you.
Thank you. Any question?
I've got a kind of hazy memory of reading somewhere that Rana viruses can be present but asymptomatic in freshwater fish populations.
Yeah.
So I just wondered how much evidence there is that it's commonly pathogenic in amphibians?
To be honest, I only focus on the disease aspect of it, but as far as I'm aware, it is pretty prevalent being a disease in amphibian populations as a pathogenic disease, causing symptoms and death. It's kind of dependent on the life stage to some extent as well. So one thing I'd quite like to do in Costa Rica is test more tadpoles. So we know in North America that die-offs are more often in these tadpole stages, and in the UK and Europe it's more often in the adults. We did have a bit of a look in the tadpoles, but didn't find any dead ones or any that looked diseased, and we didn't have the permits to kill them. So, yeah, sorry, I don't know if that answered.
Any other question? I guess just to follow up, could you imagine any study following individuals at different time scale, but at least to try to test for an effect, potential association with the...
Yeah, I'd love to obviously have long-term funding to monitor the populations. I think it's really a shame that we don't know what effect this pathogen's having, because all these species here are some of them are exceedingly highly threatened. But at the same time, we don't know even if it is having an effect. It would be lovely, it would be nice to know, or if it's potentially endemic here, or what's going on.
Okay, thank you.
Okay, thank you.
The next talk will be by Bethany Levick on Towards a Clearer Picture of the Epidemiology of Onchocercosis associated with epilepsy in the Democratic Republic of Congo.
Thank you. Well, as hinted to there by Thierry, this is obviously quite a complicated little problem, something I've been working on towards the end of my PhD. So I'll take this big, long, complicated title and focus on one bit of it to start with, which is onchocerciasis. Onchocerciasis is a parasitic infection in humans caused by infection with the roundworm Onchocerca volvulus. It's transmitted through bites of the black fly, and in humans it's fairly horrendous. So the symptoms at the extreme end include blindness. That can also range from a number of systemic infects and irritation and damage to the skin. It's also known quite well in public health as a real success story. So the two big public health programs, the onchocerciasis control program started in 1975, and then around 20 years later, the African program for onchocerciasis control. And in some areas, this has worked really very well, and the OCP in particular is estimated to have prevented around 600,000 cases of blindness. How was that achieved? Well, in the first instance, this started with larviciding. So the black fly breeds in fast-flowing river areas, and these were targeted to try and kill the larvae before they could develop into flies. Latterly, Merck have given for a number of years a commitment to provide donations of their formulation of ivermectin, mectazan even, and so that has been used particularly in that second area where it's really hard to do the larviciding because of the ecological setup of the areas, the really dense forest areas. So we move then to epilepsy, and by contrast, this is a chronic, non-communicable disease. It's caused by quite a number of factors, including some parasitic infections, and it's pretty much worldwide. I would guess that at least one of you in this room has some personal experience with epilepsy. But the thing that connects it in the first instance to the areas where we see onchocerciasis is you can see from here, the countries with the real burden of morbidity are those in sub-Saharan Africa, those really darker, redder countries. And there's various reasons for that, and that's why epilepsy has a big impact there. So, okay, they both happen in the same place, they're both causing this big morbidity burden. What brings them together? Well, over a number of years, it's been reported that there are these unusually high prevalence observations of epilepsy, particularly in countries that still have these high prevalence rates of onchocerciasis. They're hyper or holoendemic. In particular, South Sudan, Uganda, and my study country, the DRC. And essentially, the question is why? Why do we see these high prevalences of epilepsy in these areas? It's a real kind of basic public health epidemiology question, what's really causing this? So to get towards that, I've been working with a group based out in Belgium on a number of studies that are based in the northeast corner of the DRC in the Oriental province. And there's two studies that took place. So first, there was a matched case control study that was over a slightly smaller area, and then a much larger prevalence study. This recruited around just over 12,000 individuals, so it's a real big-scale study. And then within that, we've constructed a case control study by matching individuals from our database. What data did we collect? Well, in both studies, we have data about the individuals, so their height, weight, age, how many years they've spent at school. We have data about the households, who lives together, how many people there are, GPS point locations. And we also know for each individual around 10 to 15 years worth of their ivermectin history, depending upon what age they became eligible for ivermectin. You can't receive it before you're 6. In the case control study, we also were able to collect some data about people's exposure to different animals. And in particular today, I'm going to mention some things about those black fly vectors. And just to make the point, so the case control study, as I've already alluded to, is a little bit smaller. That was concentrated in this area, titulae. The prevalence study actually covers this entire geographical area. So you can see there's a real difference in scale, and even some of the localities that are included aren't on this map, so it's a very different study. We'll move to the case control study first, though. Let's start somewhere. Just to make clear, when I refer to cases, I mean people with epilepsy, not people with onchocerciasis. That's slightly different. And on my slides, I'll abbreviate that to PWE, just to make them a bit easier to read. And first case, we know that the people with epilepsy are very different individuals. They have lower height, weight, BMI, and they were the only group in which we saw these extreme symptoms of blindness and vision problems. Bringing that back to potential risks, people with epilepsy had a higher incidence prevalence of symptoms associated with onchocerciasis, such as itching and skin lesions. And actually, even when we control for their possible onchocerciasis infection by measures done in the lab, they appear to be spending more time down near these kind of fast-flowing river areas and actually at a time of day when the black flies would emerge. So it appears that they have these high rates of contact with the black flies. And pretty clearly, you can see from the graph, the people with epilepsy were receiving less ivermectin overall, and that's a significantly different relationship. So kind of we build up this picture from this first study that they're people who are probably at higher risk for the infection, are in these high-risk areas, and are getting less treatment. So we move on then to the prevalence study. This is more recent, over a bigger area, with many more individuals. And straight away, people with epilepsy appear to be getting more ivermectin, which is the complete opposite to what we were just looking at. And actually, when you put them all together in a nice little model, it's significantly, they're receiving more ivermectin overall. But actually, when you look at that in a slightly different way, so we build this case control study, and the important things here is that these individuals are matched by their location, their gender, we know this is a potential associated factor, and also their birth year. So what we're matching here is the kind of experience that they have of ivermectin dosing. It's only performed once a year. So they were kind of receiving the same experiences at the same age. And when we look at it in that manner, the people with epilepsy, again, are receiving less ivermectin. And if you reverse the question a little bit and say, okay, if I'm an individual in a given area, in a given year, and I'm eligible to receive ivermectin, what's the probability that I do so, given my epilepsy status? And when you look at the question in that sense, then the people with epilepsy are significantly less likely to receive ivermectin. And just to kind of hammer this point home a bit, when you look at the prevalence across these different areas, this whole geographical area, some of them are very high. So 8% is really getting away from being a rare disease at this point. It's relatively established. Some of them are quite low, 0.8%. And some of them are somewhere in the middle. So even across this kind of area, there isn't a consistent pattern of epilepsy prevalence. It really does seem to depend upon where in this area you are. And obviously, there might be some aspects of kind of sample size and things like that, so we can look at it in a more quantitative sense. If we take one section's worth of household data, we have the GPS point locations for them, so we'll just plot that long of them. And then we can convert that, same data, but just look at it as intensity. So across these cells, what's the probability of having the households? And we do this again, using households that only don't have epileptics. And reassuringly, they're quite similar because they should be, because 90% of the households in the left two plots are in this one on the right. So then what happens when we do the same thing, looking at households that have people with epilepsy? Well, it's actually a little bit different. So the patterns are relatively similar because the distribution of points will be sort of similar. It's one kind of location. But this patch on the right kind of is there that wasn't quite there before. Again, this is still a little bit qualitative. When you fit a model predicting the prevalence of epilepsy in a given household and include a term describing the smooth spatial distribution, it's kind of not really, it's not doing the job, it's not significant. So this little patch that appears, it's not a smooth thing, it's these individual places. So when we put all that together, it kind of tells a story that it's not kind of a big overall trend that's happening, it's very location specific. So it's to do with your ivermectin history in a particular place, and it's to do with those small, very local relationships. Realistically, unfortunately, this means it's probably quite complicated what's going on and how we do something about it. There seems to be this interplay between these kind of larger ecological factors and very divisioned up public health factors, and again, it's very much a local relationship. There will be no universal solution. One thing that is going to go ahead that I think will be really important here is some qualitative epidemiology. So you may know that even in kind of countries like our own, people with epilepsy face a certain amount of stigma and misunderstanding, and unfortunately that's only magnified in these settings. And so it may be that this qualitative epidemiology gives us some indication as to explaining people's patterns of treatment-seeking behaviour in general, as well as specific to epilepsy. And I'd just like to finish by thanking all of the people behind me and yourselves for your time. And if I can have a brief second, just to invite you all to the Parasites and Pathogens social this evening at 7. I think there's a bunch of people meeting up in the entrance at 10 too. It's at Black Lodge, which is a fantastic little pub. Thank you all again.
Thanks. So we have plenty of time for questions.
Hi.
These geographic areas, so apart from human density, what are the other sort of demographic factors affecting them? Like, is there certain areas that have, like obviously they're all relatively remote, but higher levels of a certain type of education or availability for, like, I don't know, money or any other demographic factors that would affect that specifically?
Sure. So you've nicely anticipated the exact bit of analysis I'd like to do with this. Yeah. So there are geographic differences between these areas. They've got different kind of levels of vegetation. Their kind of proximity to rivers is very different, and we anticipate that's probably going to have a factor in it. It's not just rivers themselves, it's rivers with those fast-flowing rapid areas that are permissive of blackfly breeding and emergence. So again, that's very localized. Your point about kind of more socio-demographic stuff, I couldn't say for certain. I would need to talk to the teams that go out there. As far as I understand, yes, there are differences. And anecdotally, we have some evidence of differences in public health kind of administration. So there are some areas where we have had reports that health workers don't give out ivermectin because they think it's harmful. But then there are some areas where they specifically seek out people with epilepsy because they think they're helping them, which they may well be, we don't know. So there is evidence of that, and that's definitely the next step to quantify that properly. Hi.
Is there any plan within this project to incorporate any of that vector ecology?
So not at the minute further than what I kind of discussed of the including the spatial correlates that we know are there with blackflies. I think actually one of my collaborators, Anne Lauderzo, has been collecting a number of samples because obviously it's a complex of Simulium that's transmitting, and so the different sensor stricto species that are involved we don't include for this specifically, and actually there may be some questions raised about which ones exactly are there or not, and so again, getting a proper handle on that would give some really specific detailed insight.
Any question?
No, I couldn't say. So I didn't include in here, but pretty much consistently more people with epilepsy are men, so there may be livelihood-associated factors with it. It's also, the particular thing about that one is it was before people became epileptic, and generally epilepsy appears around 12. So this is likely to be something that's happening in childhood, which maybe goes against what I said previously. So there's something happening in childhood in the way that people are interacting with the water sites then, but I couldn't say specifically why that is.
Any question? I don't. Is there treatment for epilepsy that is applied commonly, or?
There is treatment, it's not applied. So yeah, if you, or someone you knew was epileptic in this country, there are a kind of suite of drugs that could be administered and commonly are, and that are used across a number of countries in sub-Saharan Africa, but particularly some of the places here are so remote that it's been very hard because it's long-term treatment administration, so you've got to keep up that consistency, which is quite difficult in some of these settings.
So it's really debilitating in some areas?
Yeah, in some areas, yeah, particularly. And what's quite sad is that a lot of the morbidity and ultimately mortality associated with it is actually accidental. Children that have a fit and fall into a river or a fire or something like that. So it's a really different picture of what epilepsy is like compared to kind of if it was, say, in this country.
No more questions? Okay, maybe we can go to the next talk. Thank you. Yeah, you can use this, and this doesn't really work. Feel free. So, Clara Van Link will talk about consistent individual differences in immune gene expression in the wild, searching for immunological personalities and challenging the Th1, Th2 trade-off.
Yes. So hello, I'm Clara. I'm a postdoc working here at the Institute of Integrative Biology here at the University of Liverpool. And I'm going to tell you a bit about consistent individual differences in immune gene expression and hopefully explain my slightly controversial title. So for many years now, immunologists have known that different inbred strains of mice show consistently different immune responses. But what we don't know is whether this extends to natural populations, and more specifically, whether you get these consistent differences in immune response between different individuals in this natural environment. In the real wild world, individuals face a multiplicity of challenges continuously. They're infected with a range of different parasites and pathogens, and they have to cope with these at the same time as coping with environmental stress and with the stresses associated with finding food, with finding a mate, and reproducing. So do we see these consistent individual differences in this challenging and noisy environment? This is an important question to answer because it could have implications for our understanding of infectious disease risk and of immune dynamics in the wild. So some studies have started to try and answer this question, but they've mostly focused on single immune traits, which could be argued to be misleading, given that we know that the immune system is this complex network of multiple interacting traits. And this is demonstrated here by a very simplified diagram of which illustrates the diversity of Th or T helper cells, which have already been mentioned, but I'll also mention a bit more later on. So wild rodents are a great model system for trying to answer this kind of question as well, because they're the obvious bridge between what we've found out from the animal house and applying this into the field or into the real world. And the particular population of wild rodents that I work on is a population of field voles in Kielder Forest in Northumberland. And this particular population has been researched since the 1930s, but more extensively so in the past 15 years. And more recently, we've been able to sequence and annotate the vole genome, as well as to develop vole-specific immunological assays. And this has enabled us to identify functionally significant immune genes. So consistent individual differences in immune traits, or more specifically in our case in the expression of immune genes, can be said to exist if levels of expression are detectable from multiple observations of an individual after various other factors also known to affect expression and hence could possibly hide these consistencies have been taken into account. And so we asked two questions. First of all, whether such consistent individual differences in multiple immune traits can be found amongst wild individuals, and if so, whether there are any correlations between such traits. And I'll talk a little bit more about these correlations in the next slide. So these two questions can be answered by estimating these three different variables. Repeatability, which is the proportion of total variation, in this case after accounting for these other factors, which is explained by differences between individuals. In other words, how consistently different are individuals. And then there are these two different correlations which we looked for. So we thought that there might be a between individual correlation between these immune traits. And this would exist if there was a correlation between the mean value of two immune traits. So for example, an individual with a certain level of one immune trait might tend to have a certain level of another immune trait. And this would be a between individual correlation. In the behavioral ecology literature, this is often referred to as a syndrome. But another type of correlation which could exist is a within individual correlation, which occurs when, rather than mean values for different immune traits being correlated, it's the changes in these traits that are correlated. So to put this slightly differently, a change in a certain direction of one immune trait might be associated with a change in a certain direction of another immune trait. So it's this correlation between changes, just to reiterate that. So we collected data in Kielder Forest, as I said, which is in Northumberland, as shown by this red dot here. So we collected data between 2008 and 2009. This is longitudinal data, so mark recapture data, across 4 different sites. Individual voles were tagged to allow this longitudinal data set to be established. And then both on initial and subsequent capture, a range of different measurements were taken. So a blood sample was taken from the tail in order to measure the constitutive expression levels of 3 immune genes. Information about host biometric data was also collected, including information about life history stage, sex and body condition. And finally, ectoparasitic burden was estimated on visual inspection, and the presence or absence of microparasites, including Bartonella species, Babesia, and cowpox virus, was identified using PCR techniques on the peripheral blood samples that I already mentioned. And so the 3 immune genes that we focused on were IFN gamma, IL-10, and GATA3. So we know from previous studies that IFN gamma and IL-10, both cytokines, are co-expressed with functionally related genes, which in the case of IFN gamma suggests that it's involved in a wider Th1, T helper cell type 1 response, which, as we heard earlier, tends to be associated with an anti-microparasitic response. Whereas for IL-10, our previous data suggests that this has a role in the wider T regulatory response. And finally, for GATA3, we also have data from previous studies to suggest that it's associated with the Th2 response in this case, or the anti-macroparasitic response. And so, because we were interested in these consistent individual differences, we subsetted our dataset to only include those 60 individuals which were sampled at least 4 times. And we constructed a multivariate mixed effects model in which we inputted the expression levels of these 3 immune genes as the response variables. And these were all log transform standardized and assumed to have a Gaussian distribution. But before we ran this multivariate model, we ran separate univariate models to find, for each of these immune genes, to find the best set of predictors to explain the variation in these immune genes. And this best set was then used as the fixed effects in the multivariate model, and include, from left to right here, infection with Bartonella, Babesia, cowpox, as well as sex and site. And so after accounting for these variables, these predictor variables, the repeatability estimates that we got were relatively low, as you can see, in the range of 0.2, 0.3. So about 20% of total variation explained by differences between individuals. And this seems very low, but it's actually comparable to previous studies which have looked at single immune traits. And if you look at the 95% credibility intervals, none of these include 0, which suggests that all 3 immune genes, or the expression of all 3 of these immune genes, are consistently consistently repeatable. But we were still slightly concerned at these low estimates, So we decided to generate p-values by randomly permuting the dataset 1,000 times. And if you look at these p-values, we actually only have evidence for IFN gamma expression being significantly repeatable. So I think what we have is evidence for at least one, if not all 3 of these immune genes, or the expression of these 3 immune genes, being significantly repeatable, or in other words, being significantly, consistently different between individuals. We also didn't find a significant between individual correlation, so no correlation between the mean values of immune traits, but we did find a positive within individual correlation. And this is illustrated, between GATA3 and IFN gamma, and this is illustrated by this cartoon plot here. So if you were to plot the GATA3 and IFN gamma expression levels of all individuals across all of their sampling points, you would see an overall, our results suggest that you'd see an overall correlation between GATA3 and IFN gamma expression. But rather than an individual's points being clustered at one end of the slope or the other, you would actually see, our results suggest that you would see an individual's points scattered across this line, or more specifically across their own individual line, which has a very similarly positively sloped. So basically, an increase in GATA3 expression seems to tend to be associated always with an increase in IFN gamma expression. And this is actually provides no evidence for the Th1 Th2 trade-off, which we heard about again earlier. And unfortunately, I can't talk about it in this talk, so I don't have enough time, but I'm very happy to take any questions at the end. So why mention personalities? Well, the term personality has generally been reserved for the behavioral ecology literature to describe consistent individual differences in behavior, both over time and across different situations. And whenever consistencies in physiological traits are mentioned, this is usually under the umbrella of coping styles, or referring to the pace of life syndrome hypothesis. But more and more studies are suggesting that behavior is significantly dependent on physiological processes. So physiological processes could be said to equally contribute to an individual's personality, especially if we use a more general definition of a personality, as here, which suggests that a personality is simply a combination of characteristics or qualities that form an individual's distinctive character. So perhaps these consistent individual differences in immune response could be referred to as immune personalities. Anyway, I hope I've shown you that there is some evidence to suggest that there are these consistent individual differences in multiple immune traits, which could, for example, be referred to as immune profiles in natural populations. And whether or not we refer to these as immune personalities, I think is a matter of taste. So what could be the reason for these consistent individual differences in immune response? Well, as I mentioned earlier, we try to account for as many parasites and pathogens that we know infect the voles as possible, but one challenge with working with a wild data set like this is that it's never possible to include all of these. So it's possible that these consistent individual differences were the result of chronic infections, for example, that we didn't account for. The most obvious example of this perhaps would be worm burden, which we weren't able to measure in this longitudinal data set. But actually, if you remember a few slides back, our data actually shows most support for repeatability, for significant repeatability in a marker of the Th1 or anti-microparasitic response rather than the Th2 anti-macroparasitic response, which you would tend to associate with worms. So other differences that might exist between individuals which might drive these consistent differences are differences in genetics, as in the case of the lab mice that I mentioned right at the start of my talk, and also there might be some differences in the early environment that individuals experience. And the relative contributions of these, I think, are an interesting question, is an interesting question in itself. So just to give you just to briefly mention what we're planning to do next, we also know that individuals experience significant change as they go through life. So this is both in their abiotic and their biotic environment. And so the question we're trying to, so this could have consequences for these immune profiles or these consistent individual differences. So for example, a vole, as it goes through life, is increasingly exposed to its environment. And so the question we're trying to answer is whether this cumulative environmental effect might be having some effect on this immune profile and making individuals more or less individual as they go through life. So that's all from me, just to say thank you, just to mention that many other people were involved in this work, not just me, and to thank NERC for funding this work and the Forestry Commission for allowing us to conduct our fieldwork in Kielder Forest.
Thank you very much, Clara. Questions? Yeah.
My understanding is when people have looked at the relationship between gene expression and protein concentration, that's often a very poor correlation. So I wonder whether actually you might have a very strong immune personality, but the signal is being lost because you actually are measuring gene expression rather than the functional molecule. Because the functional molecule.
Yeah, that is a possibility. I think we've shown in previous studies that there is, in the field voles or in related species, there's quite a good relationship in certain cases. But I know that it's not perfect, and obviously we are looking. Sorry, I don't remember exactly, but I realise that that could be an issue, definitely, that we're looking at the mRNA kind of level, and there is that gap between the functional and the expression level. So it's something to consider, definitely.
So the question then is all about function, but of course these molecules might be slightly different sequences, which might be alleles. So you've got genetic variation at these loci as well, because that might, so the concentration of the molecule might be compensated for by different alleles.
So yeah, I didn't obviously present any of that in this talk, but I've started to look at differences basically in variations in genetic code and the effect of that on the expression levels of these immune genes, if that answers your question. Well, partly. Yeah.
Yeah.
I just want to comment on this association between Th1 and Th2 expression. Something that we have observed with humans is that persons with a healthy immune system actually tend to have a balanced expression of Th2, Th1, and Treg cytokines.
Okay, yeah, that's interesting.
So what population is that? Is that a Western population?
Yes.
Do they have no worms? No.
Yeah, the worms. Yeah, well, I know that some, obviously, it's kind of a generalization, so some parasites and pathogens also can activate both arms of the immune system to a certain extent, so it's not maybe as simple as that, and people have been debating the trade-off as well. So yeah, that's interesting. Thanks.
I'm afraid we have to stop here. Sorry. Yeah. Thank you again, Clara. And last but not least, Romain Garnier will be talking about short and long-term dynamic of antibodies against Henipa viruses in a captive African fruit bat population.
Thank you, and thank you all for staying that late. So I'm going to talk about a research project that I am doing in a captive bat population in Africa that's part of a postdoc fellowship funded by the AXA Research Fund. And I don't think I'm going to need to convince anyone in the attendance that bats are actually important reservoirs of zoonotic disease. So if you want to name just a few of the last 20 years, Endra in Australia, Nipah in Southeast Asia, but SARS, MERS, Marburg, and maybe more recently Ebola are all related in some ways to bats, and bats are generally considered reservoirs for all of those viruses. So just, for instance, the latest West African Ebola is potentially originating from a virus thought to have been zoonotic transmission from a bat to a 2-year-old boy in December 2013. I would take that sentence with a tiny bit of salt because it's still being debated, but that gives an idea of how important bats actually are. Some of the problems, though, is that, so they have all those viruses, but we have very, very limited evidence of die-offs in bats, with the only exception being the white nose syndrome, so the very recent fungal infection of bats that have been killing them very well. But viruses killing bats, rabies, but that's hardly a die-off. The others, pretty much no evidence of die-offs. And there's a very strong, that's been published quite recently, support for a coevolution between the bat immune system and the bat and viruses in bats from genomic studies. But we still have a very limited understanding of the mechanisms at play in the bat immune system, and we only know that for a few species of bats, and the bat is one of the most diverse group of mammals on Earth. For instance, the studies of innate immunity, so pattern recognition receptors or the interferon response, have shown that there's a lot of functional differences between what we know of model mammals, so humans, or small rodents, and bats. They seem to be working slightly or importantly differently. The problem is we really don't know much about acquired immunity, and one of the problems is we have little access to captive bats, and recapturing the same individuals in the wild is close to impossible. Just to give you an idea, the program, the people I work with have been working a program in wild bats in Africa, and in one location, 3,000 bats have been tagged. The recapture rate of that is about 1 to 2%. So recapturing the same individual and monitoring the same individual is pretty much impossible. So the idea in 2009 was to start using a captive colony in which bats would actually reproduce. The species chosen was the one you can see right here, the African straw-colored fruit bat, so Eidolon elvum for the scientific name, that are actually naturally infected with Nipah viruses. So the same family as Endra, the Australian one, or Nipah in Southeast Asia, but an African version of it that's quite vaguely known only since it's never been isolated. Only genetic material has been found. Interestingly, they also don't have filoviruses. The captive ones don't have filoviruses, and they don't have rabies. Very useful when you need to work with them in terms of biosecurity. So where do we keep those bats? Well, we keep those bats in that kind of structure. So that's a massive cage that has a 27.5 meter diameter. So they can actually fly in it, which means they're in perfect weight and as close as they can be from being in a natural kind of condition. The colony was established in 2009, a total of 77 individuals, and early 2016 the colony was about 125 individuals, with very few remaining from the original individuals. We also have a second facility about 100 meters from where that one is. They both are in Accra in Ghana, so still in West Africa, that allows isolating bats for experimental infection work, but also for easier access. Some results that have been found in the cage, that's a paper by Kate Baker published in 2014, and what they found is that there's a seasonal dynamics of anti-Nipah viruses antibodies in those bats, with females, not males. Males don't show any kind of dynamics, but in females, there's around March, that's in 2 consecutive years, around March, there's an increase in antibody levels, but the time scale between blood sample was about 2 to 3 months, so very difficult to interpret further than saying that there might be some seasonal dynamics. So the kind of question I want to ask today is, first of all, how variable is the acquired immune response, but over a shorter period of time? So having a better handle on those dynamics. But also, now we have data from 2009. What can we learn from that long-term monitoring in terms of the dynamics of viruses in a small captive population? So I'm going to first start by that short period of time dynamics. What I did is isolated 16 individuals in groups of 4. They are much better than if you keep them alone. Between early March and late May, so 12 weeks isolation. Supposedly around time of parturition, they were expected to give birth in April, but ecology doesn't work exactly as you want, so they didn't give birth during the study period. And weekly, taking a blood sample and weigh the bats and analyze the blood samples for antibodies against Nipah viruses. We have Luminex, so that's basically a bead array that allows to measure specific antibodies against Nipah viruses and a bunch of others. So we call that, it's called the mean fluorescence index, because the antibodies attach to the microbeads, and then you throw a laser at them, and depending on whether there's an antibody attached or not, then you get a value that's correlated with how much antibodies was in the original serum. So what we get is that kind of things on the 16 individuals. So in other words, the short-term antibody levels of mostly gravid female, there's a male hidden in there, in the negative one at the bottom, highly variable. And if you throw a linear model at it, you actually find that there's a cubic relation with time. So that's by far the best model here. I think that the early dip that you find in that data, that's quite obvious between... Does that actually work? Vaguely. So the first 2 weeks of data show that early dip, and I think that might be an artifact due probably to the stress of having taken them from that 125 strong population and isolated them in groups of 4. So I don't want to over-interpret that one. The late dip, though, could be due to some things going on in very late pregnancy. So by the time we had to release them for ethical permit reasons, most of the females were starting to go down, and most of them were actually about to give birth at that point. So obviously, need more data later on, but yeah, that's one thing. The other thing that's quite interesting is here you only have the positive individuals, and that's the relation between the mean antibody level and the weight. And what you find is that all the slopes are actually positive with weight, all of them. And the question is it just weight, or is it some kind of more specific condition going on? So it would be very interesting, obviously, on those samples to have access to more protein physiological measures, so albumin total proteins. So that's a step that's I'm actually working on at the moment. So stay tuned, next time maybe. So clearly, the other thing is, again, a follow-up after birth would be very interesting, because bats have a very, very specific challenge. They can't put on too much weight, otherwise they lose their flying weight. They can't fly anymore. So the condition of bats at the end of pregnancy is probably quite bad in the females, which could have some impacts on immunity as well. So from here, I'm going to move to the second part of that talk about monitoring, long-term monitoring and dynamics of viruses. So the captive colony, we have actually some decent sampling between 2009 and 2016. And we have this same Luminex technique has been used since the onset of the project. So we have the entire time series of it. The colony size has been variable, but always below about 130 individuals. So what that means is that the colony size is definitely insufficient to sustain the transmission of a virus. If it's directly transmitted and transmission is occurring all the time, that would be way too small a population for that to happen. So if we find evidence of recent transmission, then it's more likely that the virus is never actually eliminated and some individuals can stay infected for a very long period of time, which would be very interesting in the debate that there is in the bat viruses community, whether bats actually get infected, eliminate, and the virus keeps going on, or bat actually never eliminate and the virus can hide and then resurface at later dates. And what we find is, so we have 14 time points. Be careful of the x-axis because the time points are not the same. So there are some actually wide gap of like between January 2012 and January 2014, with 2 years where we don't have data analyzed yet. But for now, what you have in front of you is 14 time points, 223 individuals, which include the original 70 strong abouts. 964 sample, but we also have another 600 that we are currently analyzing and that will come to complement and basically bridge that gap between 2012 and 2014. Just for clarity, those are the individuals on which we have the most data, so 19 individuals that have been sampled more than 8 times. And we can see a variety of dynamics going on here, variety of things. Just to make it simple, and I've taken out like 3 of the very typical things that we see. The first one is some individuals just keep their antibodies, so that's the green line that you can see, so always somewhat high, never very high, but somewhat constant. Some others show some sort of boosting, so that's the red line, so it first goes quite well down, then back up, then completely down, and then back up again. And the last one, well, that one is very interesting because that one is actually a new infection, and that's this new infection occurred at least 2 years after the population was established. So some evidence of new infections as well. Further evidence of new infections can be seen here in looking at individuals that we caught in March 2016, so 7 years after the colony was established. And the blue ones are actually individuals that were already tagged in March 2016. And the green ones are individuals that we tagged at that time. So those are born in 2014, 2015, so completely new, born in captivity. And we find a lower prevalence, sure, but we do find a prevalence. So that means that some individuals born very recently got infected, which, yeah, goes very, very clearly towards there's some kind of resurgence of virus in the cage at some point. One thing that I think would be very interesting is to better estimate the seroconversion rate in the captive populations, and that data set would be perfect for capture-recapture studies, multi-event capture-recapture, and estimate transition rate between the different categories. And on that, I'm just going to finish by thanking all the people we work with. So at Cambridge, James Wood and Olivier Restif, but also in Ghana. So Richard Sawyer at the Forestry Commission especially is very important for that work. And again, thank you very much for your attention.
Thank you. Short time for question.
Can I just ask about the assay? How specific is it for this particular virus, and how do you know it?
It's obviously not completely specific because we don't have the African Nipah virus. It's never been isolated. So we use a cross-reaction with Endra and Nipah. So what I'm...
So that's your antigen bait in your assay,
basically, the antigen is fixed on a bead. And what we use, we actually have 4. I'm only showing 1 because all 4 of them correlate fairly well. We have Andra G protein, NIPA G protein, Andra F protein, and NIPA F protein. And all four of these, the F proteins generally respond not as well as the G, but the Andra G and the NIPA G are doing fairly well.
And is it one isotype of antibody in the bats, or is it multiple isotypes?
We don't really know because we don't have a specific secondary antibody for bats. So we are using biotin. So we have to use strepavidin. So we have to detect whatever binds. So probably some mix of IgG, IgM.
Thank you. It's more or less time to finish. Well, thanks to all talks for all the talks, and I guess a lot of us can meet tonight at the social event about parasites and pathogens.